So in our experiment, we have two cell types that we're using, MLOI4s, which are osteoblasts, which build the calcium onto the bone. And we also have the MC3T3s, which are the osteocytes, which are kind of like mission control. They're communicators that tell what the other cells to do, whether they need to take calcium off of the bone or put some more calcium back on. So that's what we're working with, and we're going to have osteocytes and osteoblasts alone to see what happens, and then we have a co-culture of those two together to see if they're communicating, if that makes a difference. So these are our MLOI4 cells, and they are really confluent, which means that there's the whole bottom of the flask is covered in cells. They're at about 90% confluency, and once you have too many, it gets too crowded. So you have cells that are competing for food and they're competing for space, so they'll start to die. So we want to thin those out. So we have all these flasks right here that we need to thin out, but we don't need this many cells. So what we're going to do is we're going to take out about a third of them and add them to another flask, and then we're just going to have to bleach the other ones, because if we just keep going, we'll have like 100 flasks and we don't need that much. So right now we have the trypsin, which is going to it's going to break the bonds that the cells have to the bottom of the flask so that they'll release, so that I can get them out of the flask. But that has to be at 37 degrees C before I can use it. This is what the MLOI4s, or the osteoblasts, look like under a microscope. You can see how crowded they are at the bottom of that flask, and all the white dots are cells that have died and have lifted off of the bottom of the flask. This is what the MC3T3s, or osteocytes, look like under the microscope. You can see how crowded they are, so they're definitely ready to split so that we can seed them onto cytopore and put into the plate for our experiment. Once we have enough cells, we put them onto cytopore beads. That's the beads that you're seeing under the microscope, the spheres. And on each of those spheres, we have either osteoblasts, osteocytes, or a co-culture of the two of those. Once the cells are on the cytopore beads, we can then put them into the 96 well plate to look at the fluorescence of the dye inside the cells that's either bound or unbound to calcium at 405 or 485 nanometers.